All right, so we're going to do a video here on cellular reproduction. You're going to want to go to your Hapara workspace again. I'm just going to use first hour here, cell reproduction. So I'm going to go ahead and open up the notes for the cell reproduction, and we're going to go through them. When we talk about cellular reproduction, we're talking about two different processes. Really, we're going to focus in on mitosis and meiosis. But what you can look at here is when we talk about cell reproduction or cellular reproduction overall, we're talking about the whole process of growth and division. And the division part really is just this small piece of the pie right here. As you can see, over 75% of the cell reproduction cycle for most cells is in this growth phase. And then we go through the division phase, which is either mitosis or meiosis. It can be broken up, the growth part can be broken up into three phases. G1, which is your major growth, cell growth during the S phase or synthesis phase. That's where we have the DNA replication happening that we talked about when we were in the DNA notes. And then G2 can be a little bit more growth, and it is also preparation for cell division. As you can see right here, the G1 or gap 1 phase happens right after cytokinesis, but before DNA synthesis. It's where your cell, whatever kind of cell it is, this is where it does most of its functional thing. Is it a muscle cell? Is it a bone cell? Is it a stomach lining cell? Whatever it may be, it's going to do its job within this skin cells, okay? Most cells spend their time in G1 performing whatever is required. So whatever their job is. The S phase, right there in the middle, is the synthesis phase. It is where your DNA is replicated. Your chromosomes are going to divide to form into identical sister chromatids, and they're going to be connected by the centromeres. So it does take a little bit of time to do that DNA replication. Then finally, we have the G2 or gap 2 area, and you have twice as much DNA as you did in G1 because of S phase and that it duplicated. So the chromosomes begin to shorten and coil up and get ready for division there. So this is what interphase looks like. You still have your nuclear membrane intact, your chromatins inside. You have the nucleolus inside the nucleus. And you have your centrosomes or paired centrioles kind of still at one end. When we talk about mitosis, all right, we have some prefixes here. And these prefixes are very important to kind of remember. And then you might want to add some stuff here in notes or in a PDF creator. But pro means before, and that's really our starting point. Meta means after, or I like to use meta as middle. Ana is away or onward, and telo is end or tello. Now, the thing that you got to understand when we talk about mitosis or meiosis is we're asking, where are the chromosomes located? Are they sister chromatids? Are they chromosomes? What are they, and where are they located? Now, we're going to go ahead and get into some different things. Mitosis, again, is part of cell reproduction. Two identical cells are made from one cell. This is how we produce body cells. In the process of mitosis, we start with one cell and we get two new identical cells, which we call diploid cells, meaning they have a full set of 46 chromosomes. We can put those into pairs, and we have 23 pairs of chromosomes when we talk about human beings, but they have a full 46 chromosomes. A new cell also can be referred to as a 2N cell. Some of the major players in mitosis is the cytoplasm. Well, you'll hear me talking about that. Nucleus, nuclear membrane, chromosomes, centrioles, and centromeres. We go through mitosis due to the need for body growth and repair. We also look at it having 4 stages: prophase, metaphase, anaphase, telophase. And then after telophase, we have cytokinesis, which is not on here, which is the splitting of the cytoplasm. And then we also have interphase, which is not considered to be part of mitosis, but it is a part of the overall cellular reproduction. Some special cases in mitosis: nervous tissues are limited in mitotic divisions. We call them apoptotic or apoptosis. It cannot go through multiple divisions. So, for instance, brain tissue and spinal cord cannot repair itself or regrow after it's damaged. So we look at prophase as being the first stage in mitosis. We see the chromosomes becoming visible. Nuclear membrane starts to disintegrate, also with the nucleolus. Your centrosomes, which are the paired centrioles, they start to move to the opposite ends and start to form what we call spindle fibers in between them. Some of the fibers are going to start to attach to the chromosomes in the late prophase area. And they start to begin to pull the chromosomes toward the equator of the cell. During late prophase and into what we call pro-metaphase, there's 2 events that kind of happen. You see the nuclear membrane totally gone, it disintegrates, and the attachment of the chromosomes to the spindles. As you can see right here, these would be your centrosomes, or your paired centrioles, kind of starting to move to the opposite ends. The chromosomes are condensing into that sister chromatid look, where they're held together by the centromeres. And then you have your nuclear membrane starting to break down. In metaphase, like I said, I like to have M stand for middle. You have the double chromosomes, or the sister chromatids. They're attached to the spindle fibers by their centromeres. The chromosomes are pulled by that spindle fiber to the middle, or the equator of the cell. And your spindle fibers have extended from one pole to the opposite pole. So as you can see right here, this equatorial plate, or the metaphase plate, right here in the middle of the cell, you have your sister chromatids that are connected to the spindle fibers, and now they are lined up right in the middle of the cell. Anaphase, the A meaning standing for away or apart. Anaphase is marked by the separation of your sister chromatids. Now they are just chromatids, and they are starting to be pulled apart by the shortening of the spindle fibers. So as you can see here in the image, you have the chromosomes have split, and they're starting to get pulled to the opposite ends where the centrosomes are. Telophase is the last phase in mitosis. We have a set of chromosomes in each end. The chromatids are at each end, and what we see is the new nuclear envelope starts to form around each set. The double membrane begins to form between 2 nuclei. So as you can see, you're starting to have them at the ends. You would have nuclear membranes starting to come back together. And then this, what we call a cleavage furrow, starts to pinch in the new 2 cells to lead us into cytokinesis, which is the splitting of the cell cytoplasm. In the animal cell, what we see is the cell membrane pinches in along the equator. In the plant cell, we have what we call a cell plate that gets put across the equator and divides the cell into 2 new cells. So that's the process of mitosis. Again, we use those for body growth and repair when we have issues with some of our cells that need to be repaired quickly due to some damage. We have cells replacing those cells all the time as they wear out naturally. The biggest example that I like to talk about is your stomach lining cells. Due to the amount of acid in the stomach cell, we are constantly replacing. We're constantly replacing those so that we can continue to update what's going on there. Now, meiosis is how we reproduce our sex cells. So an egg cell in the female and a sperm cell in the male. What we do is we start with one cell and it results in 4 what we call haploid cells, or cells that have half the number of chromosomes. Remember, a diploid or diploid cell for a full set, haploid cell for a half of the chromosomes. Again, it's the same parts that are involved in mitosis: the cytoplasm, nucleus, nuclear membrane, chromosomes, centrioles, and centromeres. Really, the big difference here is we have 8 stages in meiosis: prophase 1, metaphase 1, anaphase 1, telophase 1. Then we do the whole section again because, again, we need to split it in half. So we would have 46 chromosomes. We need to split that in half again. So we go through a second series that's called prophase 2, metaphase 2, anaphase 2, and telophase 2. And like it says here, sometimes you would skip straight from anaphase 1 to metaphase 2, skipping telophase 1 through prophase 2. Interphase, again, is not considered part of mitosis because it is part of cell reproduction. So the big thing that you're going to see is a lot of these stages look the same. It's just we have some more breaking down happening. The big phase that's different in meiosis and mitosis is that this prophase 1 through telophase 1, we are looking more at a crossing over area. This prophase 1 crossing over is when we have double chromosomes. The sister chromatids kind of pair up in what we call a tetrad, which would be 4. And what we have is we have same areas of chromosomes switching places. You can kind of see it on the colors here. I'm not even going to try to guess what the colors are, but you see identical parts being switched. So what this crossing over allows for is information not to be necessarily identical. Each sister chromosome still has the information for the trait carried on that chromosome, but what we do is we get a big time variation there. Okay, so during prophase 1, everything is the same. Okay, you're gonna have your nuclear membrane breaking down. You're gonna have your centrosomes moving to opposite ends of the poles. The spindle fiber is going to form towards the late prophase, early metaphase 1. You're going to see the tetrads being attached to the spindle fibers, and then as you can see here in metaphase 1, again, M stands for middle. We have the tetrads of homologous chromosomes attached by the centromere to the spindle fibers. Homologous meaning same, so those chromosomes are going to kind of line up together. Then we get anaphase, and what you see now that the tetrad has split in half. Now we have the double chromosomes, or the sister chromatids, move to the opposite ends, being pulled by the spindle fibers to the centrosomes. Looks very similar to everything that happened in mitosis. Again, in telophase 1, into cytokinesis, they're not in sister tetrad, or they're not in tetrad form anymore. Now they're in sister chromatids. They have gotten to the ends, and you start to see the nuclear membrane kind of reforming around the sister chromatids. It will enter interphase, or it can, or it may skip it. Okay, if it needs to do a little bit of growth, it might grow again. But the organ, but it won't go through S phase because the initial amount of chromosomes in there. So again, we have them at the 2 ends. We would have 2 cells split here. So now we're in the prophase 2. We have 2 cells. They're not identical in the sense that they're the same genetic layout, but they do have the same number of chromosomes here, okay? Occurs just like prophase in mitosis. So prophase 2 through telophase 2 is almost identical to mitosis. Your sister chromatids are going to start to coil up, condense, and what they're going to do is they're going to attach to the spindle fibers that, again, the centrosomes have moved to opposite ends. The spindle fibers are forming in between them. We're going to get our sister chromatids to attach there. In metaphase 2, again, they're going to line up right in the middle, attached by the centromeres. It's going to hold the sister chromatids together during anaphase 2. Again, A still stands for being pulled apart or away. Occurs just like in mitosis. We have the chromosomes being pulled to opposite ends. Okay. The double chromosome split and each chromatid moves to the end of the cell. And then during telophase 2, just like in mitosis, the chromosomes uncoil, nucleolus, nuclear membrane starts to reform, and then cytokinesis, we end up with 4 individual haploid cells. So as we look to compare and contrast mitosis, meiosis, we have this whole thing of cell reproduction. They share in common. I could have put interphase in here also. Sister chromatids are in both. But the big thing is we have 4 stages to 8 stages. The diploid or full set of chromosomes versus a haploid or half the number. The type of cell being made, we have a somatic or body cell in mitosis. What we give the name for meiosis is gamete or sex cell. What our result is 2 identical daughter cells for mitosis. And over here in meiosis, we have 4 daughter cells with half the chromosome info. And the big thing difference is in meiosis, we have tetrads. And that's where our crossing over happens. So hopefully you will spend some time reading through this, listening to the video a couple times. Watch Hank's video on mitosis, meiosis, and you should be good.